Aerobic respiration, oxidative phosphorylation. Oxidative phosphorylation, or OP, is the final process of aerobic cellular respiration. The energy from the electrons harvested from the previous steps is used to set up a proton concentration gradient across the inner membrane of the mitochondria. Then the gradient is used to power the production of ATP molecules. So far, a lot of work has been done to achieve a net gain of 4 ATP molecules, but chemical potential energy has been stored in the NADH and FADH2 molecules. The big energy payoff is in oxidative phosphorylation, where the energy stored in those molecules is used to create somewhere between 26 and 28 ATP molecules. So in our overall cell respiration process, we're over here in the last step now of oxidative phosphorylation, which consists of 2 parts: electron transport chain, ETC, and chemiosmosis. And take a look at what it is coming into this process. It's electrons via NADH and FADH2. There's a short movie you can click on to download here to see what happens as a result of the electron transport chain. We're going to go through the steps in detail in some slides up ahead, but I do want to read through the text version of the video narrative just so you get an overall overview of electron transport chain. So the electron transport chain is a series of protein complexes embedded in the mitochondrial membrane, which you can see in the image, all of these protein complexes. Electrons captured from donor molecules are transferred through these complexes. So we talked before about those electrons coming from previous steps in cell respiration here. And when they do, they're transferred through each of these protein complexes. Coupled with this transfer is the pumping of hydrogen ions. This pumping generates the gradient used by the ATP synthase complex, which we see here, to synthesize ATP. The following complexes are found in the electron transport chain: Nadh dehydrogenase, Cytochrome BC1, Cytochrome Oxidase, and the complex that makes ATP, Atp Synthase. In addition to these complexes, 2 mobile carriers are also involved: Ubiquinone here and Cytochrome C here. Other key components in this process are NADH and the electrons from it, hydrogen ions, molecular oxygen, water, ADP, and the phosphate groups, which combine to form ATP. At the start of the electron transport chain, 2 electrons are passed from NADH into the Nadh dehydrogenase complex here. Coupled with this transfer is the pumping of 1 hydrogen ion for each electron. And as I mentioned, we're going to talk about that in detail in a few slides. Next, the 2 electrons are transferred to ubiquinone here. Ubiquinone is called a mobile transfer complex because it moves the electrons to the Cytochrome BC1 complex here. Each electron is then passed from the Cytochrome BC1 complex to Cytochrome C here. Cytochrome C accepts each electron one at a time. 1 hydrogen is pumped through the complex as each electron is transferred. The next major step occurs in the cytochrome oxidase complex here. This step requires 4 electrons. These 4 electrons interact with a molecular oxygen molecule and 8 hydrogen ions. The 4 electrons, 4 of the hydrogen ions, and the molecular oxygen are used to form 2 water molecules. The other 4 hydrogen ions are pumped across the membrane. This series of hydrogen pumping steps creates a gradient. The potential energy in this gradient is used by ATP synthase here to form ATP from ADP and inorganic phosphate. The ATP synthesis steps you see here are discussed in greater detail in the ATP Synthase Gradients animation, which is also linked throughout this presentation. This animation illustrates 2 full cycles of electron donation. In biological systems, however, many electron transport cycles occur simultaneously, helping to ensure that the proton gradient is always maintained. There's also a video linked here that in detail covers the chemiosmosis process that occurs at the ATP synthase portion at the end of the electron transport chain. In the event that you're unable to download and view the movie, I'm going to record the movie narrative from the text version that you can listen to and use this still image from the video to provide some context. Again, in the coming slides in this section, we're going to go into each of these steps in more detail, but I do find that the narrative from the video is helpful. Concentration gradients are a key component of the biological world. The potential energy from these gradients is often used to perform biological work. Here, we focus on hydrogen ion concentration gradients. Hydrogen ions are also known as protons. That's why in the image you see high proton concentration or low proton concentration. A gradient exists when there is a higher concentration of a molecule in one compartment compared to a neighboring compartment. This animation will demonstrate how the potential energy that results from a hydrogen ion gradient uses ADP and inorganic phosphate, also known as PI, to synthesize ATP. So we're taking the energy stored from this concentration gradient. That stored potential energy is used to form the chemical bond between ADP and phosphate. Notice here the large difference in the number of hydrogen ions on the 2 sides of the membrane. This difference is a hydrogen ion or proton concentration gradient. The energy associated with this gradient is used to synthesize ATP from ADP and inorganic phosphate. This occurs at the ATP synthase complex shown here. 1 hydrogen ion enters the ATP synthase complex from the intermembrane space, and a second hydrogen ion leaves it on the matrix space. The upper right part of the ATP synthase complex rotates when a new hydrogen ion enters. Once 3 protons have entered the matrix space, there's enough energy in the ATP synthase complex to synthesize 1 ATP. So, in other words, hydrogen ions flow from here into the ATP synthase, and once 3 of them are there, then we can make ATP. Let's take a look now at these processes in more detail. And again, if you can click on and watch the video, it is very helpful to see it as an animation rather than just hearing the transcript. So do so if you are able. One thing to note here is that in biological systems, the gradient is always maintained. So one thing that happens in the animation is that eventually the gradient, as these hydrogens move across, the process completes and stops because we no longer have a high proton concentration and a low proton concentration. But in biological systems, that gradient is always maintained. The mitochondrial hydrogen ion gradient is generated as electrons pass through the 3 membrane complexes in the electron transport chain that we talked about previously. All right, let's take a closer look at the electron transport chain. The electron transport chain involves a movement of electrons that requires the presence of oxygen. NADH and FADH2 store energy. There's energy stored here, and that energy can be released in the form of high energy electrons, but they cannot do it if oxygen is not available. Oxygen is an electron acceptor. The high energy electrons cannot flow unless they have a final destination. Oxygen is that destination and will take the electrons. High energy electrons donated by NADH and FADH2 molecules. So you can see that happening as NADH becomes NAD plus, FADH2 becomes FAD plus. You see these high energy electrons. They move in the direction of electron flow here across the inner mitochondrial membrane. And once they get over here, I'm just following this yellow arrow, they are de-energized here, okay? As they flow through the electron transport chain, they're using up their energy, and over here, they're accepted by oxygen through the series of reduction and oxidation reactions that occur. So we'll talk more about what happens here at the end, but just see that these high energy electrons are being produced here, traveling through the electron transport chain, and are accepted in the mitochondrial matrix by oxygen. As the high energy electrons move down the membrane, so in this direction through the electron transport chain, they're able to transfer their energy to proton pumps. Proton pumps do just what they do. What it sounds like they do, they pump protons or move protons across the inner mitochondrial membrane against the proton concentration gradient. So it moves them from an area of low concentration to an area of higher concentration. And you can see that the reason for that is for them to be used later. The continued movement of protons results in more protons outside the membrane than inside, resulting in a net positive charge outside the membrane and a net negative charge within. The outside protons are attracted to the inside, but the membrane only allows them to move out. ATP synthase provides a channel back to the inside, and the protons diffuse through the ATP synthase molecule in the semi-permeable membrane through facilitated diffusion during chemiosmosis. That final movement of the protons uses their electric potential energy of their gradient across the membrane to add a phosphate group to an ADP, creating ATP. Energy from that electrochemical proton gradient is converted to chemical energy. The electron transport chain and chemiosmosis is the same process we discussed in photosynthesis. There's a video here for ATP synthase that I'm going to show now.
Concentration gradients are a key component of the biological world. The potential energy from these gradients is often used to perform biological work. Here we will focus on hydrogen ion concentration gradients. Hydrogen ions are also known as protons. A gradient exists when there is a higher concentration of a molecule in one compartment compared to a neighboring compartment. This animation will demonstrate how the potential energy that results from a hydrogen ion gradient uses ADP and inorganic phosphate, also known as PI, to synthesize ATP. This process involves an enzyme complex called ATP synthase. Gradients and the potential energy they create are key aspects of the biological world. A good example of the use of a gradient occurs in the mitochondria when ATP is synthesized. ATP is synthesized by ATP synthase, a large complex of membrane-bound protein. Here we see ATP synthase, along with other membrane-bound proteins. Notice the large difference in the number of hydrogen ions on the two sides of the membrane. This difference is a hydrogen ion, or proton, concentration gradient. The energy associated with this gradient is used to synthesize ATP from ADP and PI. This occurs at the ATP synthase complex. One hydrogen ion enters the ATP synthase complex from the intermembrane space, and a second hydrogen ion leaves it on the matrix space. The upper part of the ATP synthase complex rotates when a new hydrogen ion enters. Once 3 protons have entered the matrix space, there is enough energy in the ATP synthase complex to synthesize 1 ATP. In this way, the energy in the hydrogen ion gradient is used to make ATP. Now let's watch the process again. Notice how the proton enters the ATP synthase and exits into the matrix space. Once 3 more hydrogen ions have crossed the membrane, another molecule of ATP will be made. In this example, the hydrogen ion gradient is large enough to produce 6 ATP molecules. Please watch as the remaining ATP molecules are synthesized. The process has now completed, and the result is an equal number of protons on each side of the inner membrane. Without a gradient, there is no more energy available to make ATP. In biological systems, however, a gradient is always maintained. The mitochondrial hydrogen ion gradient is generated as electrons pass through 3 membrane complexes. That process can be seen in the mitochondrial electron transport chain animation.
The electric potential energy from the protons is used, along with ATP synthase acting as a catalyst, to add a phosphate group to ADP, creating ATP. Electric energy, or the potential electric energy from the gradient, is converted to chemical energy. Finally, the de-energized protons here and electrons here are taken by the electron acceptor, oxygen, making water. Counting the molecules. The molecules present after the complete breakdown of a glucose in aerobic respiration is 30 to 32 ATP, carbon dioxide, 0 NADH because they've been oxidized by the electron transport chain, and 0 FADH2 for the same reason. These figures are lower than the previous agreed upon estimate of 36 to 38 ATP molecules from aerobic respiration. The current estimate now takes into account the energetic cost of shuttling ATP into and out of the mitochondria and the NADH made in glycolysis into the electron transport chain. The ATP output varies between 30 and 32 ATP due to the variable efficiency of the two processes. The production of 32 ATP molecules assume an efficient shuttling mechanism, while 30 ATP assumes an inefficient one. But where do the 30 to 32 ATP come from? Here is a breakdown. In glycolysis, the net energy output is in the form of 2 ATP and 2 NADH, which result in a final ATP output of 2 ATP or 3 to 5 NADH. Pyruvate oxidation produces 2 NADH, which gives a final ATP output of 5 ATP. The Krebs cycle or citric acid cycle has a net output of 2 ATP, 6 NADH, and 2 FADH2, which can result in 2 ATP, 15 ATP, and 3 ATP respectively, for a total of 30 to 32 ATP. Proton uncoupling and thermogenesis. The electron transport chain generates a proton gradient across the mitochondrial inner membrane. ATP synthase uses the electric potential energy from the proton gradient to synthesize ATP from ADP, a process we've discussed in detail and shown here in A. Proton uncouplers have a similar process, but the electric potential energy is transformed into heat and not used to synthesize ATP, which you see in image B. So rather than this series of protein membrane protein complexes in ATP synthase and ATP synthase, we have a proton uncoupler that instead converts the electric potential energy to heat. The production of heat is called thermogenesis. Mitochondrial proton uncoupling is one of the dominant mechanisms through which endotherms, or warm-blooded organisms, regulate their body temperature.